Hello everyone, my name is Cayleen. I work here at the CRIPS lab at SDSU, and today I'm going to tell you about why I heart flies. So my research focuses on congenital heart disease using Drosophila as a model organism. A congenital disease is a defect that is present at birth. And as you can imagine, a congenital heart disease is going to be a defect that is in the heart at birth. Now, there are various types of congenital diseases, and out of all of these, congenital heart disease is the one that occurs the most, and this happens in one out of every 3 infants that have some form of congenital disease. This isn't something that just affects children or infants, but it also affects adults, and 1 out of every 150 adults has some sort of cardiac defect. Now, as I mentioned before, I work with the Drosophila model system, and Drosophila is just a fancy name for a fruit fly. And unlike humans who have a 4-chamber heart, Drosophila actually have a long cardiac tube. Now, you might wonder why we would study heart development in humans when the heart of a fly is so different, but they actually have very similar developmental mechanisms, and so a lot of great research has been done to show us how the human heart develops. We're going to leave our fly friend over here for a minute and step over to this side. And so we know that in our genome we have DNA that codes for the proteins that our body needs in order to function properly. Now, there are other proteins that are called transcription factors that make sure that this DNA gets expressed at the correct time. What I work with is a protein called Tin Man. And if your initial thought is, hey, that sounds a lot like the character from The Wizard of Oz, you would be exactly correct. Because if flies cannot make this protein, just like our friend the Tin Man, they do not have hearts. And so what Tin Man is, it's a transcription factor that has several domains within it. And the domain that I look at is the tin domain. And so what I'm looking at is what role does this tin domain play in the ability of the Tin Man transcription factor to be able to interact with the other proteins that are needed in order to get expression of the protein that we want. I also want to know if I change this domain and cause a mutation, what sort of phenotypic effects do we see in the hearts of flies? You might ask, why do we study a protein that's in flies? And the reasoning is because for proteins that are very important to development, many species have similar versions of it. So in flies, we have the Tin Man, and in humans, we have NKX 2.5. And this also has that same tin domain. And so by getting insight into what the tin domain does in the fly Tin Man protein, I can get further insight into into the important role that the tin domain plays in the human protein NKX 2.5. Thank you for listening to the type of research that I do, and I hope you now understand why I heart flies.